Hi, this is Raja, and I'm going to present on Staphylococcus aureus food poisoning. So, I will start my presentation with an article I read few weeks ago about food poisoning. So, food poisoning may seem insignificant, but it has big impact in some areas, such as aviation industry. So, in 1982, on a flight from Boston to Lisbon, 10 crew members of a flight got food poisoning, including pilot, co-pilot, and flight engineer. The flight was able to return around and land back in Boston without further incident. After that incident, airlines made an official rule stating that pilot and co-pilot cannot eat the same thing on a flight. So let's talk about Staph aureus. Staphylococcus aureus is a gram-positive, round-shaped cocci bacteria present in the form of pairs, chains, and bunched grape-like clusters. It is also called golden staph and Orostaphyra. It is non-spore forming, facultative, anaerobe, which means it can grow without oxygen, and it belongs to Staphylococcus genus, which is divided into 32 species and subspecies. It can survive in high salt and low water activity habitats, and it is a catalase and coagulase positive. Epidemiology. Staphylococcus was first isolated from pus from an infected knee joint by Surgeon Alexander Oxton in 1880 in Scotland, and it was given the name Staphylococcus aureus by Frederick Julius. It is commonly found in soil, water, and air and can be found in human nose, throat, hair, skin, and as a normal flora of lower reproductive tract in females. Normally it is harmless but can cause infections as it is an opportunistic pathogen. Approximately 30% of the population are the carrier of Staphylococcus aureus. It can cause multiple infections and is one of the most common causes of nosocomial infections. Each year, 500,000 patients in the United States get infected with Staph aureus and cause up to 50,000 deaths. Pathogenic strains of Staph aureus can cause infections by producing toxins and express cell surface protein that bind to antibodies and inactivate them. So, Staph aureus food poisoning is one of the most common forms of food poisoning in the United States. It can produce an enterotoxin called staphylococcal enterotoxin, which is responsible for food poisoning. There are different types of enterotoxin produced by Staph aureus, and type A of enterotoxin is commonly related to staphylococcal food poisoning. Other types of enterotoxin produced by Staph aureus are SEB, SED, EH, G, and I, and all of them can cause staphylococcal food poisoning. So different strains of Staph aureus produce a different quantity of enterotoxin during their exponential phase of growth. Our recent research shows that as low as 1 microgram of enterotoxin can cause food poisoning. Genes responsible for producing enterotoxin are located on plasmids and prophages, which are mobile elements, and can be transferred between strains of Staph aureus, modifying the ability to produce enterotoxin and cause illness. The optimum temperature of Staph aureus to produce enterotoxin is between 40 to 45 degrees centigrade, but the temperature could be between 10 degrees centigrade to 48 degrees centigrade. The production of enterotoxin is highly dependable on the temperature, so low amount of enterotoxin will be produced with the decrease in temperature. As the production of enterotoxin depends on temperature, it is observed that enterotoxin by itself is highly temperature resistant. Enterotoxin remains stable in frozen condition and it is highly resistant to heat. It can also survive the sterilization process used in canned food. As Staph aureus is facultative anaerobe, the production of enterotoxin can occur in both aerobic and anaerobic conditions. However, aerobic conditions are most suitable for toxin production. Toxin production also depends on pH conditions. The optimum pH range for Staph aureus to produce enterotoxin is 7 to 8, with a wide range of 4.5 to 9.6. Staphylococcus food poisoning is not caused by the bacteria itself, but instead the enterotoxin produced by the bacteria is the main culprit. Low pH and high heat can be used to eliminate Staph aureus, but the enterotoxin are highly resistant to these conditions. Enterotoxins are also resistant to proteolytic enzymes, and because of that, all these characteristics, enterotoxin is able to survive in the gastrointestinal tract after ingestion. The size of enterotoxin ranges from 22 to 28 kDa, and in their structure, at top of N-terminal, there are the disulfide loops, which are flexible in nature and makes enterotoxin more stable, and also associated with the induction of vomiting caused by enterotoxin. So once ingested, enterotoxins are able to penetrate in the intestinal tract and target the immune system of the host. Enterotoxin is a superantigen and it makes the host immune system to produce a large amount of histamine, which causes vomiting. It also inhibits the absorption of water and electrolytes in the small intestine and can cause diarrhea. Mode of transmission. So, food poisoning caused by Staphylococcus aureus is also called staphylococcal enteritis. Staph aureus will produce enterotoxin when it is allowed to grow on typically protein-rich food for several hours. The foods which can cause staphylococcal enteritis include cream-filled bakery items, eggs, meat salads, meat, pudding, and sometimes vegetables. Enterotoxin can present on the food which shows no sign of spoilage. Food can be contaminated by the food handlers who are the carrier of Staph aureus. Now the symptoms. Symptoms of Staphylococcus food poisoning generally appear within few hours, in between 1 hour to 6 hours, after the ingestion of enough amount of enterotoxin which can cause food poisoning. Common symptoms are nausea, abdominal cramps, weakness, diarrhea, fever, and explosive vomiting, which can retain for a few hours. Fatalities due to Staph aureus food poisoning are rare. It's around like 0.03% for the general public, but are occasionally reported in young children and the elderly, which is 4.4% fatality rate. Staphylococcal food poisoning can cause to the people of all ages and it depends on the amount of enterotoxin ingested by the host. However, young hosts and elderly will develop more serious symptoms. And in lab diagnosis, a stool culture may be required for isolation of Staph aureus. Samples of food can also be tested. There are several rapid tests like ELISA and reverse passive latex agglutination available for the detection of enterotoxins. Most of the time, lab protocols are not needed as a physician can diagnose food poisoning easily by the clinical symptoms. Now, the treatment. As, as the staphylococcal food poisoning is caused by the enterotoxins, So no antibiotics are needed in this situation. Medicines can be given to the patient which can control the vomiting and nausea. Plenty of fluid intakes is recommended for the patients to avoid dehydration. Intravenous fluids can be administered in severe dehydration. Soft foods are recommended during the food poisoning, and foods with high fibers should be avoided as they are hard to digest. Illness can last up to 1 to 3 days and it will be resolved by itself. So, now the questions. So, question number one: Staphylococcus food poisoning is caused by which type of toxin? Endotoxin, enterotoxin, shigatoxin, or shigatoxin? And the answer is enterotoxin. Question number two: Staphylococcus aureus is a facultative anaerobe. True or false? The answer is true. Question number three: Staphylococcus aureus can be present in soil, water, nose, hair, or all of the above. So it's all of the above. Question number four: Which of the following is not a symptom of Staphylococcus poisoning? Vomiting, nausea, diarrhea, and petechia. And the answer is petechia. Question number five: Which antibiotic is used to treat staphylococcal food poisoning? Penicillin, cephalosporin, tetracycline, or no antibiotic needed? So the answer is no antibiotic needed. So that's it, and thank you. So if you have any question, you can send me a message on Canvas, or maybe you can comment down below. Thank you.